Hello everyone and welcome to Armstrong's Laboratory. On this channel, we take academic papers and break them down into simpler terms so the typical layperson can understand the material. In this video, we are breaking down the paper, Ribosomal DNA transcription in prefrontal pyramidal neurons is decreased in people who killed themselves. This paper investigated how gene expression is impacted in the brains of individuals who killed themselves. You can find a link to the paper I am discussing in the description below. A key area of interest for these researchers is the prefrontal cortex. The prefrontal cortex is the part of the brain that is responsible for complex cognitive behaviors such as modulating intense emotions, considering the future and making predictions about it, foreseeing and weighing possible consequences of one's behaviors, and inhibiting and initiating appropriate behavior. I plan on making a more in-depth video regarding the prefrontal cortex at a later date, but for now, just remember that this brain area is of specific interest for this paper. Now, take a moment and think back to your high school biology class. You probably remember your teacher talking about the central dogma of biology, or DNA to RNA to protein. DNA is the genetic carrier of information and encodes for all of the genes in your body. For instance, DNA encodes for ribosomes, or the machinery that is responsible for synthesizing proteins. Ribosomal DNA, or rDNA, houses the information for the synthesis of ribosomes. When rDNA is clustered together, this is referred to as a nucleolar organizing region. Nucleolar organizing regions represent locations in the cell that are being actively transcribed, or having their DNA being processed into RNA. When stained with a silver dye solution, scientists can detect levels of transcription occurring from rDNA. This is accomplished by assessing the area, number, and ratio of silver staining to unstained areas of the cell. We have to take a minute to ask ourselves what the purpose of rDNA plays in cells. Currently, rDNA is thought to play a role in brain plasticity. This is specifically referring to the plasticity of neurons, a type of brain cell that is responsible for electrochemically communicating with other neurons. Plasticity can be defined as the ability of the brain to change its structure and function in response to stimuli. rDNA has been shown to have lower levels of gene expression within parts of the brain, such as the prefrontal cortex, within individuals who have killed themselves. Less rDNA expression may lead to altered brain plasticity. The scientists in this paper hypothesized that this decrease in rDNA is caused by oxidative stress seen specifically within pyramidal neurons. Now, pyramidal neurons are a specific type of neuron that have a single axon with many dendrites. Oxidative stress is the imbalance of free radical activity and antioxidant activity. Free radicals are unstable atoms that are missing an electron, while antioxidants are substances that inhibit oxidation or the loss of electrons. An antioxidant example can include things such as vitamin C. In this paper, the scientists took portions of the prefrontal cortex from both individuals who killed themselves and individuals who did not kill themselves. The prefrontal cortex was sectioned into four specific parts: the dorsolateral prefrontal cortex, the ventral and dorsal anterior cingulate cortex, and the orbitofrontal cortex. The researchers then investigated 8 regions from cortical layers 3 and 4. They then stained these sections of the brain with the silver dye solution. With these sections of the brain and the silver staining, they were kind of able to determine the transcriptional activity of rDNA in these brain sections. The results show that there appeared to be less of a silver dye stain in the brains of individuals who killed themselves compared to the control group of individuals who did not kill themselves. This was only seen when comparing the area of the stain between groups, as the number of stains and the ratio of stained regions to unstained regions did not differ. Interestingly, they found a lower silver staining in male subject brains compared to female subject brains. Now, some literature out there has shown that there are fundamental differences in brain function between male and female brains from individuals who have killed themselves. The study itself was limited in how many female brains they had available to them, so I would take their data with a grain of salt. However, this does open up the door for looking into sex-related differences in individuals who killed themselves. So the scientists in this study show decreased rDNA expression through silver staining. Let's take a look at what factors impact rDNA gene expression. The main factor impacting rDNA gene expression is the mTOR pathway. And many other things can impact the activity of the mTOR pathway. For example, a decrease in glucocorticoid receptors, the receptor that is responsible for processing stress, can impact the mTOR pathway. This lines up with the current literature showing decreased glucocorticoid receptors in the prefrontal cortex of suicide victims. In addition, these glucocorticoid receptors can lose their function by the presence of a polyamine, or a positively charged molecule that interacts with negative proteins such as the glucocorticoid receptor. In addition, mTOR signaling pathways can be impacted by decreased gene expression of SCA2, which is involved in the glucocorticoid receptor's transport from the cytoplasm of neurons. Other studies have shown that SCA2 is implicated within cell division as well. I'll be making a more in-depth video about SCA2 at a later date. Another factor potentially impacting mTOR signaling is glutamatergic transmission in the prefrontal cortex. This refers to the neurotransmission of glutamate, a neurotransmitter that makes it more likely for neurons to communicate with each other. In the brains of individuals who have killed themselves, a blockage of glutamate receptors on GABAergic neurons may lead to decreased activity of the mTOR pathway. GABAergic neurons are neurons that primarily contain GABA, a neurotransmitter that makes it less likely for neurons to communicate with each other. In suicide victims, research has shown that there is a hyperactivity of GABAergic neurons. This hyperactivity may decrease mTOR activity by suppressing BDNF, or brain-derived neurotrophic factor. BDNF is a protein activator of the mTOR pathway. So with decreased expression of BDNF, this would result in decreased mTOR activity and thus decreased rDNA expression. Research has also shown that decreased BDNF may be impacted by the expression of serotonin. This paper also discussed the role of microglia in individuals who have killed themselves. Microglia are brain cells that are involved in the immune response. These cells may be inducing an oxidative stress response in various pyramidal neurons. Oxidative stress may be induced by a specific serotonin receptor known as 5-HT2A. Both microglia and this receptor may be decreasing mTOR activity through a specific protein known as RED1, a potent mTOR inhibitor. Finally, the paper discusses the possible role of methylation as potentially impacting rDNA gene expression. Methylation is the addition of a methyl group to a DNA strand. When the methyl group is present, it acts as a physical barrier for the machinery that transcribes DNA into RNA. Ultimately, this results in less rDNA gene expression. As we've seen, many things can impact the expression of rDNA, from the mTOR pathway, glucocorticoid receptors, SCA2, glutamatergic transmission, and GABAergic neurons, oxidative stress, and methylation. All of these factors may be playing a role in decreasing rDNA gene expression, which ultimately impacts the brain plasticity of individuals who kill themselves. Thank you for taking the time to watch this video. I hope you enjoyed it and learned something new. If you have any questions, please leave a comment down below and I'll be happy to answer it. If you enjoyed this content and want to learn more about mental health, please subscribe to the channel and give this video a like. I plan on doing weekly videos in which I release more information about mental health. Thank you again for watching. I hope you learned something.